So briefly with this slide, I just want to introduce you to medicinal inorganic chemistry, which is the branch of bioinorganic chemistry in which our group mostly works. So this is a broad area of medicinal chemistry, which comprehends a number of applications of inorganic elements and compounds in medicine. And some of these applications, you can see them listed here. So, for example, even those of you who are not related to biological bio-oriented research might have heard the story of platinum compounds as anti-cancer agents. So we have methyl compounds which are therapeutic agents for different diseases, and the platinum family became particularly important in the treatment of cancer, and is still nowadays one of the main chemotherapeutic regimes. But then you can also have compounds that are instead have been used as diagnostic agents, for example, for MRI, radiopharmaceuticals, either for diagnostic and therapeutic applications. And then, of course, there is the field of chelation therapy that uses organic molecules able to sequester endogenous metal ions that in certain disease states have been let's say put out of their regular homeostasis. And this is, for example, the case of Alzheimer's disease treatments by chelation therapy, etc. So we are talking about endogenous and exogenous metal ions in principle in this area. So during my career, I've touched upon several of these sub areas. And throughout, I think since my postdoc, I really focus on the therapeutic agents. So in this slide here, I just want to give you an overview of our research interest. So of course, we are interested in developing inorganic drugs, methyl-based compounds for therapy and imaging, but we want this compound to be highly targeted. So for example, we want to develop these complexes as targeted protein inhibitors for specific families of proteins, which are relevant to the disease of interest. And mostly we worked on compounds which have anti-cancer effects, anti-proliferative activity, but we are also interested more recently in antibacterial agents or antiprotozoal agents for the treatment of different parasitic diseases. And of course, our research path in this area really resembles and corresponds to the one of classical medicinal chemists who are dealing mostly with organic small molecules, drug-based therapeutic agents. And so we are always in the search of structure-activity relationships, but to have such SARS, we need to understand the mechanism of action of our methyl compounds. And so a great deal of our efforts is spent in the mechanistic studies, which are addressed by a very integrated and interdisciplinary approach. We start from biophysicals and biological methods to more pharmacological type of assays. So we arrive in principle to perform when a compound is interesting in a series. We can arrive to perform all the preclinical phases of investigation up to the animals. So normally we run animal studies in collaboration with other groups. When I was in the Netherlands, I was in a pharmacy institute, so my group also learned how to do animal studies. But for the moment here at we arrive at the cellular level. And so it's our chemistry, our approach, investigational approach is very similar to the one of classical medicinal chemists. However, we have an additional challenge. And the challenge here is that our methyl-based compounds can undergo extensive speciation in biological environment, in aqueous environment. That's the medium that we are dealing with. That's our solvent, of reaction solvent. And so speciation, which is different from the one of organic compounds, from their metabolism. This is a speciation that includes, for example, ligand substitution reactions with biological nucleophiles, or even redox processes. And so it's absolutely essential for us to control this methyl speciation. We want to benefit of it because eventually we want our compound to, for example, bind a biological target via ligand exchange reaction. However, we want that this reaction occurs exactly in the time and space that we want, so at the target level, not before, not when the compound is injected in the bloodstream, not before it enters the cell, and so on and so forth. And so a common strategy to try to achieve this control on the methyl speciation that the medicinal and organic chemistry community has taken since a number of years already has been the use of and the development of organometallic compounds, because with organometallics we have stronger methyl carbon bonds that somehow allows us the fine-tuning of this reactivity. And very recently, in the last 6-7 years, some of us have decided to try to capitalize on the well-known catalytic properties of methyl compounds, including organometallics, and to try to achieve what we call catalysis in cells. I will give an example of this approach in one of my case studies if I have time. So unfortunately, not all our best targeted compounds, for example, we have compounds that are extremely good as inhibitors of a selected protein targets. But this inhibition is, which is ideal in vitro, in a solution that contains only the target and the compound. This doesn't mean that then the compound can become a drug lead, because maybe there are also toxicity effect, or the pharmacokinetics is not right. So what to do with these perfect inhibitors, perfectly designed inhibitors that then are not they cannot be translated in further steps of the drug discovery? We can still use them as a selective inhibitor, exactly, to understand the function, the roles of that specific target in the biological context. For several targets, we have some proteins are, for example, associated to cancer development, but it's still not clear how, through which pathways. And treating cells with our inhibitors allows to selectively quench the function, the activity of that protein, and see the resulting effects on at the cellular level. And in this sense, our compounds are chemical probes that can help us elucidating the role of certain targets in a disease mechanism. And from the perspective of this disease mechanism, so the knowledge that we are interested in achieving, this is important, and this can be done in different ways, either via the use of these methyl-based chemical probes that I just mentioned, or, for example, and this is more a problem of bioinorganic chemistry, is really to try to achieve a better understanding of the role of endogenous metal ions in the mechanism of disease. For example, in cancer development, in cancer cell proliferation and invasion. And I just mentioned here a project that we have ongoing with a network of laboratories worldwide on the understanding of the role of copper ions in the spread of cancer. So there are different reports that somehow are controversial and sometimes highlight how the presence of this metal ion in cancer cell is actually beneficial to the cancer cell growth. Other reports instead are evidencing a toxic effect, specific again to cancer cells. So we are trying to figure out how this works, and this is really one of the fundamental bioorganic chemistry questions that we are conducting in our group. So a few years ago, before I joined TUM as professor, but I was Hans Fischer Senior Fellow at the Institute for Advanced Studies, I had the possibility to start a new project, not this time involving small molecules, metal-based drugs, but more complex metal-based entities, specifically supramolecular coordination complexes. And with these supramolecular coordination complexes in mind, we started to develop bioactive materials, particularly for applications in drug delivery. So as novel drug delivery systems or as novel theranostics. So systems that are endowed with both therapeutics and diagnostic properties, which some metal ions-based systems are endowed with. And so this experience in the development of bioactive material then led us also to join forces with other scientists here in our department, including Roland Fischer, including Bernard Rieger, to put together a new project that has been recently funded by TUM, an innovation network on the design of smart supramolecular materials for regenerative medicine. So we are really at the beginning of this project, but we capitalize on our experience on these other drug delivery metal-based systems. And finally, the last point which we are very interested in is actually the importance of non-covalent interactions in our chemistry and in our compounds or supramolecular coordination complexes reactivities. So of course we want that, for example, in the case of inorganic drugs, our compounds arrive to target a protein and form a covalent or pseudo-covalent coordination bond, a stable bond. But we have also realized that to make a compound selective and with high affinity for a biological target, there are a number of non-covalent interactions that takes place at the very beginning, you know, of the process of interaction, and this can highly alter the selectivity of our compound. And these include pi-pi stacking interactions, electrostatic interactions, hydrogen bonding, and so on. On the other hand, the same non-covalent interactions have high importance. They're essential to drive the process of host gas chemistry of our bioactive materials, where we want to encapsulate, for example, a host drug molecule based on these purely non-covalent interactions. So we are really challenged by these aspects here, because it's not easy to study these interactions with classical methods, or at least we don't have high throughput methods to evaluate and give a real weight to the influence of these interactions. Anyway, the next slide here is just here to help me showing you some of our typical targets. Well, I've been talking a lot about proteins, but which proteins? So we have proteins that are already binding to metal ions. For example, zinc finger domains that bind zinc. And these are also relevant domains in proteins that are related to cancer spread. And so sometimes we want to alter with our synthetic metal-based drugs, specifically this metal binding domain. Other proteins instead are completely not dealing with metal ions on a regular basis, but they are again important targets for the disease of interest. So this is the case of membrane proteins, water channels, for which we have developed already 10 years ago one of the most selective inhibitors that you can find in the literature. And also we have other type of targets like nucleic acids, and hopefully I will show you, to talk about this in the next slides. So I've selected just 3 case studies for you to have an idea. The first one is about the possibility to do one day maybe catalysis in a cellular environment. And for the moment we are at the level of performing gold templated bioorthogonal reactions in physiological conditions. This is where we are now. So the metal, the selected metal in this case is gold. And we have discovered recently a peculiar reactivity of our gold compounds with protein targets, which allows to perform what we call bioorthogonal modifications of our protein systems. Bioorthogonal because the reaction per se is not existing in living systems, and thanks to our metal center, we can actually have this reactivity. And for those of you who are a little familiar with gold in catalysis application, not bio application, but generally in catalysis, it's well known that gold is an excellent Lewis acid that is able to activate, for example, alkyne bonds towards nucleophilic addition. And there are a lot of examples of organic transformation in the literature that uses gold as catalyst with this specific idea of Lewis activation in mind. However, in our case, reacting a specific scaffold, organometallic scaffold, these gold-3 cyclometallated complexes with a bidentate CN ligand. So reacting this scaffold with a protein containing cysteine residues, the first step is a binding step, direct binding step of the gold center to the thiol of a cysteine residues that is relatively soft and has affinity for gold binding. And so this is quite a classical metal protein adduct. However, with this specific family of complexes, the reaction could proceed forward, and via a process that is well known in catalysis, named reductive elimination, we could achieve a cross-coupling reaction templated by the metal center. Cross-coupling that led to the formation of a new sulfur-carbon bond. And you can see here that the ligand has been of the original metal complex has been completely transferred to the sulfur of the cysteine. So this reaction is occurring in few minutes in physiological condition, meaning pH 7 aqueous solution. So we have, since this first report, we have started investigating the mechanism of action. We have been characterizing structurally via different methods, also calculations, the different intermediates that we thought were relevant for the reactivity. And this picture here just summarized you that we are a bit obsessed with the understanding of how these compounds work with biomolecules at the molecular atomic level. And here it's a picture that we obtained via QM calculations, but as a matter of fact, this picture has been validated by a number of other experimental techniques. And this is just the summary of some of the steps of this reaction. So in the future with this project, because we have been able to achieve a selective permanent modification of an amino acid on a protein, then we want to use our compounds for proteomic profiling of cysteine residues in biomolecules. This is very relevant to protein target identification, protein tagging, protein purification. So there are a lot of implications that this reactivity in a complex cellular environment can allow us to achieve. And we are actually developing already some preliminary step in this project with the group of Stefan Acker, Liebig Fellow in the Sieber group. And eventually we hope that we will be able to use our compounds as therapeutic catalyst, selectively modifying one or very few cysteines in a whole proteomic pool of a cancer cells or a bacterial cell. But for the moment, until we arrive to the therapeutic catalyst, we are actually trying to capitalize on the mechanism that we have learned for the carbon-sulfur cross-coupling to see if we can achieve in mild condition or even in aqueous environment, starting from the same type of compounds, other types of cross-coupling reactions. And here it's a paper that came out last year where we proved that this concept is also valid for carbon-phosphorus cross-coupling reactions. This is not biologically relevant at the moment for us, but that tells us that from the fundamental knowledge of the bioinorganic mechanism, we can actually arrive to to have some influence in other areas of chemistry and catalysis in the future. Certainly to understand better the steps of the catalytic processes at goal 3. I don't know how much time I have, but talking about the non-covalent interactions that can be established between metal-based compounds and biological targets, I present here a case of bis organometallic compounds, basically an heteroleptic bis NHC compound, which is not prone to ligand exchange reactions like the other family of goal 3 compounds that I show you. These two bonds here are extremely stable, so the compounds tend to stay intact in the cell. And this led us to study the interaction of this small molecule with a particular family of biological targets, specifically the DNA G quadruplexes, which are guanine-rich regions of DNA that can fold to form a specific secondary structure of DNA. It's not the classical B structure, but specifically the G4 structure. And so here we had to understand these non-covalent interactions and to quantify the binding energy of different compounds in our family, starting from this first one, which is depicted here. We had to move towards advanced atomistic simulation. And in this case, this is a video that is obtained via metadynamic simulations that at variance with classical MD methods allows not only to obtain a nice video, but also to calculate exactly the free energy surfaces that describe the binding strength of these compounds in the different poses that you see. That we don't put any bias in this movie. We just let the compound interact with the molecule, and you can see it can go away and can come back and then find a stable binding site at the top of this G quadruplex structure. So of course we the simulations and the atomistic view picture of the interaction is important, but on the other hand, we have to always remember that we are interested in understanding the overall mode of action of this compound. And for this specific derivative, we recently studied via proteomic approaches the effects of the complex in cancer cells, in ovarian cancer cells. And this paper that was out in Chemistry European Journal last December, I can tell you is one of the most dense that I have produced so far, with a lot of co-workers and collaborators. But the interdisciplinary aspect of this research demand us to be extremely to involve several other people in this story. So I just have a few minutes, so I will go just to the last example. So this is about supramolecular coordination complexes for biomedical applications. It's where we started entering the supramolecular chemistry domain, but always with metals. And in this case, our supramolecular coordination complexes are actually discrete entities. If you are immediately thinking of MOF, these are the, let's say, the equivalent of MOFs, but not polymeric in nature, but with the same, built up with the same principle. So via self-assembly of coordination of mixtures of molecular building blocks, metal ions and ligands, okay? And the idea for us was to exploit the possibility of these metalla cages, supramolecular entities, to establish host-gas interactions, specifically host-gas interactions with a drug or with an imaging agent. And just to show you, this is one of the latest examples that we achieved. We have proved in this paper that not only the host gas complex is stable in vitro, but also in vivo. And it can encapsulate a radioactive isotope, pertechnetate, and then via injection in mice, this host gas complex can even arrive, if appropriately targeted, so bioconjugated to peptidic domain, it can even arrive to the brain of the mouse. And so here it's a long way to drug delivery and theranostic agents development, but it's another fascinating area, and we are learning a lot via different techniques on the possibilities to tune these entities for our biological purposes. I just end, I won't talk about the last new projects, but I just end thanking my group members. This was an old picture of my group taken in 2019 that I still need to update. I hope in the summer this will be possible, but 4 people, four people actually are still with me. You have already met some of them. Darren is taking care of the computational part. Sophie Thomas and Riccardo Bonsignore, they are all more on the synthesis. Breck Eichmann, she's wrapping up with her PhD thesis, but she's our cell expert. And Claudia Schmidt, she's a new postdoc in my group, and she joined us early last year, but she has already 2 or 3 projects managing among those that I already showed you. So, and thank you very much for your attention.